over the inner ear. The inner ear anatomy, when we look at this piece right here, the center piece right here is called the vestibule. This piece over here that looks like a snail is the cochlea. And then these three right here are the semicircular canals. So that is basically the inner ear. But when we talk about the inner ear, there are three different chambers. Let me write out semicircular canal. Now, we say that we have two osseous labyrinths and an inner or middle layer, which is membranous labyrinth. So the word labyrinth literally means a maze. So if we were going to look at the vestibule, the center portion, you have a, let me grab a different color pink. You have a center layer right here that's called the membranous labyrinth. Now, there is a slightly smaller swollen piece right here of the membranous labyrinth in the vestibule and a little bit larger one right here, but the membranous labyrinth is the middle chamber. Now, the membranous labyrinth is in all three areas. So it is all connected. And then whenever you talk about the semicircular canals, there's going to be a slightly swollen portion of the membranous labyrinth on each semicircular canals. But literally, each piece is going to be connected. This isn't a perfect drawing, but basically we can talk about the three chambers of each area. So we're always going to have a superior chamber and an inferior chamber, and then the middle chamber is called the membranous labyrinth. Now, there are different names for the swollen areas inside of the vestibule. In the vestibule, you basically have an area here that we're going to talk about. This is a special area, and then there's going to be three special membranous labyrinth here, and then this one has its own name. So when we talk about the inner ear, this is the vestibule. This is the cochlea, and this is the semicircular canals. However, when we look at the inside, we are going to go into more anatomy, and we call this the labyrinth. So the labyrinth is always going to be three chambers. Now, the outer portion that you see in this brown color is bone. So the superior and inferior layer is always known as the osseous labyrinth. And then the middle layer is going to be known as the membranous labyrinth. So when looking at the vestibule, we're going to use this as basically our model. We can say that the top portion is bone. So I'm just going to draw it like this. And the bottom portion is bone. So when we talk about the labyrinth inside, the superior aspect where it's bone is the osseous labyrinth, and it has a name. And think about the vestibule as the centerpiece. The superior chamber is called the scala vestibuli. This is literally the superior chamber. Now, the membranous labyrinth is created by a membrane. So even though I have it written out right here is just all pink, literally the membrane that is underneath the scala vestibuli that divides it into the three layers, this is called the vestibular because it is under the scala vestibuli membrane. So there's another layer that divides, this would be like the top region, and then this is going to also have another membrane right here, and this is known as the basilar membrane. Think of a basement being on the bottom. So together, these membranes make up the membranous labyrinth. Now, labyrinth means maze. So we have this inner chamber the middle chamber, which is made by the vestibular membrane and the basilar membrane, and we literally call this the membranous labyrinth. Labyrinth again means maze. It's the middle layer. Now, every layer in the vestibule, the cochlea, and the semicircular canals are going to have a different name. So there's a different name for each membranous labyrinth in the vestibule, a different name for the membranous labyrinth in the cochlea, and a different name for the membranous labyrinth in the semicircular canals. So the membranous labyrinth, the membranes are going to stay the same, but we're literally just going to say a different name to describe the membranous labyrinth in each of the areas in the vestibule, in the cochlea, and in the semicircular canals. But when we look at the three chambers, so literally you have a top layer, and the top layer, whereas the vestibule, this represents bone. This is bone. So the superior chamber, this is the top. The superior chamber is always going to be called the scala vestibuli. And then the inferior chamber, which is from the basilar membrane to the bone on this side, so this is going to be the name of the inferior chamber. The inferior chamber's name is scalae tympani. Now, these are their names, but because this is bone on the outside, we literally say that the superior chamber, which is named scalae vestibuli, and the inferior chamber, which is named scalae tympani, is known as the osseous, osseous means bone, labyrinth. And we will always say that the superior osseous labyrinth is called scala vestibuli, and the inferior chamber, 1, 2, 3, which is also the osseous labyrinth, maze, it's called scala tympani. So for each region, you would say scala vestibuli, then you have membranous labyrinth, scalae tympani. Scalae vestibuli, membranous labyrinth, scalae tympani. Scalae vestibuli, membranous labyrinth, scalae tympani. But that is part of the osseous labyrinth. Now, when we discuss the osseous labyrinth and the membranous labyrinth is going to have a different name for each area, which I'm going to discuss the different names. But before we can talk about the inner ear is literally responsible for generating action potentials for hearing at the cochlea, static equilibrium at the vestibule, and dynamic equilibrium at the semicircular canals. So we're going to have these different organs in the membranous labyrinth that are going to generate action potentials. So before I can literally go into that, I need to go over the different types of fluid in the osseous labyrinth versus the membranous labyrinth. So we have two osseous labyrinth, that's the superior chamber and the inferior chamber. And so again, the bone is represented by the outside. So there's always 3 layers in each area. So scalae vestibuli, membranous labyrinth, and scalae tympani. And the scalae vestibuli and scalae tympani are part of the osseous labyrinth. Now these are going to be filled up with fluid. So when we talk about the osseous labyrinth, so we have bone and we have bone. We have the superior chamber. The superior chamber is always going to be called scalae vestibuli. Superior chamber. The inferior chamber is always going to be called scalae tympani. Now, the membranous labyrinth literally is formed by the vestibular membrane on the top and the basilar membrane on the bottom. This is the membranous labyrinth. So you need to know the names of the membranes. vestibular membrane is the first membrane, and the basilar membrane is the second membrane. This is how you make the membranous labyrinth. Now, inside of your scaly vestibule, we have fluid that is going to basically circulate on the outside and the inside. So the name of the fluid that is going to be in the osseous labyrinth, so this is going to be filled up with a fluid, which we call perilymph. So the word peri means around. So your osseous labyrinth is filled up with fluid, which I'm just coloring in green, known as perilymph. So perilymph is always going to be found inside of the osseous labyrinth in both the superior and inferior chambers, which has their own names. Now, the membranous labyrinth, which has different names in each region, also has a fluid. The fluid inside of the membranous labyrinth that is inside of the ear is always going to be known as endo. So endo means inside. Endolymph. Endolymph. This is the fluid inside of the membranous labyrinth. So this is the membranous labyrinth. Which are going to have different names in each region, and it's always going to be filled with endolymph or fluid. Now, your scala vestibule is created by bone on the outside. The scala tympani has bone on the outside, so it's 3 chambers. So perilymph is the name of the fluid inside of the osseous labyrinth. So, both the scala vestibule and the scala tympani is known as the osseous labyrinth, but they have their own names for the superior chamber and the inferior chamber. So we're going to go through and we're going to talk about each of the areas, but you need to understand this to understand how the inner ear is going to do its job. So when we talk about the cochlea, its membranous labyrinth is known as the cochlear duct. This is literally the membranous labyrinth of the cochlea. That's pretty easy, cochlear duct. Now, the vestibule has 2, and the 3 semicircular canals has 1, or 1 for each semicircular canal, and they have their own names. So the first one I'm going to talk about is the cochlear duct. Now, each of the membranous labyrinth is going to have specialized structures that are going to help us be able to hear or to help us with dynamic or static equilibrium. So I'm going to go over the cochlea. Now, inside of your cochlea, you have the specialized structure that is going to be called the organ of Corti. We're not going to draw it on here. We're going to draw out basically this with the organ of Corti. So we're on the cochlea. The cochlea is where we are. So we're in the cochlea. The cochlea is the organ of hearing. So literally on here, we say it contains the organ of hearing inside of the membranous labyrinth called the cochlear duct, but the organ of hearing is called the organ of Corti. It's purple. So the first thing that we're going to discuss are the 3 layers. You're always going to draw the 3 layers. So the top layer, scala vestibule, the bottom layer, scala tympani, And then you're going to have two membranes, which is going to make up your cochlear duct. So you have the vestibular membrane, and at the bottom you have your basilar membrane. Now, it's very important that you understand the three parts of the cochlea, because how we hear, it goes through a process which we call auditory transduction. So when we talk about the scaly vestibule, the scaly vestibule is literally going to be connected to the stapes. The stapes is an auditory ossicle that is going to basically vibrate the fluid inside of the scaly vestibule, known as perilymph. So there has to be a way for the middle ear to basically send vibrations into the ear. So when we look at the scala vestibule, the superior chamber, there's a window right here called the oval window. The superior chamber is where everything begins. So the scala vestibule is basically going to have the oval window. Now, as sound vibrations come into the inner ear, we need to vibrate the fluid inside of the scala vestibule. So your stapes, which is an auditory ossicle in the middle ear, has a foot plate that also looks like an oval. This literally is the piece of the stapes, so it looks like a stirrup. And vibrations are going to basically make the stapes push onto the oval window of the superior chamber called the scala vestibule. And it's easy to kind of think about when you say the vestibule. So scala vestibule. So vibrations from the tympanic membrane vibrate the stapes, and the stapes foot plate is going to basically push onto the oval window, which is going to vibrate the perilymph and the scala vestibule, or the scala vestibule. Now, vibrations are going to vibrate the perilymph, which is going to vibrate the vestibular membrane, which is here. So the vibrations literally travel from the stapes through the oval window into the scala vestibule, through the basilar membrane, into what we call the cochlear duct. So we need to know the different names. So the organ of Corti is a specialized structure which is in the cochlear duct. So the Membranous Labyrinth is called the cochlear duct. And the membrane on the bottom is called the Basilar Membrane. And the organ of Corti literally sits right here on top of the basilar membrane. So I'm using purple. This is your organ of Corti. Now, coming out of your organ of Corti are little hairs or cilia, which are known as sterocilia. They're little microvilli that come off the hair. So we're going to use green. Inside of your organ of Corti are little hair cells, and these hair cells have little microvilli that are floating around in the endolymph. These are hair cells. With microvilli, which are into the cochlear duct, what they're floating around in the endolymph. Stereocilia. These are the little microvilli. Now, as the stapes acts like a piston or auditory ossicle, receives vibrations, vibrations are going to travel as the stapes pushes the oval window, so vibrations. This is part of auditory transduction. So vibrations move into the scaly vestibule, which has endolymph. So the endolymph is going to vibrate. So the vibrations travel through the vestibular membrane. Now these are going to bend the hair cells, but most of your hair cells are going to bend as the vibrations travel through the basilar membrane. So the basilar membrane literally is vibrating because this is full of endolymph and you're vibrating the fluid. So auditory transduction is talking about sound vibrations going to the auricle, to the external auditory canal, to the tympanic membrane, to the auditory ossicles, which is malleus, incus, then stapes. Well, the stapes is going to connect to the oval window, and the stapes is literally touching a membrane which is vibrating the endolymph of the scaly vestibule. The scaly vestibule's vibrations are going to travel through the vestibular membrane, and then the vibrations travel to the basilar membrane. So the blue is basically representing vibrations. Sound waves create vibration. So what happens when the basilar membrane vibrates is these little hairs, these little microvilli, are going to rub onto a membrane that sits right above the hair cells. So I'm going to draw it out a little bit. The membrane that the hair cells bend to against is known as the tectoral membrane. So vibrations, sound waves, make the basilar membrane vibrate, and these hair cells bend back and forth in the endolymph against the tectoral membrane. Well, these little hair cells, when they bend against the tectoral membrane, these are going to act as like little receptors, and as the hair cells bend, they basically are going to activate the cochlear branch of cranial nerve 8. So let's write that out. Movement of the basilar membrane. Now the basilar membrane is going to move because of vibrations. They're going to bend the hair cells that have microvilli called stereocilia against the membrane, the tectoral membrane. We should say hair cells of the organ of Corti. The organ of Corti is literally the organ of hearing because it has the hair cells. The organ of Corti is how you hear. So the movement of the basilar membrane bend hair cells of the organ of Corti against the tectoral membrane, and as the hair cells bend against the tectoral membrane, this is literally, these hair cells are connected. They're going to stimulate the cochlear branch of cranial nerve 8. Now, of course, we've already learned the brain. So from the brain, or it's going to go to the thalamus to be processed, and then it's going to go to the temporal lobe. So literally, the organ of Corti is the organ of hearing. And the way that you are going to stimulate the organ of Corti to stimulate cranial nerve 8 is these hair cells bend and then electrical impulse travels down these hair cells to cranial nerve 8. They bend against the tectoral membrane. So, when we say auditory transduction is always going to have to go through the entire process of the ear. So, when looking at the anatomy of the ear, it has to go through the auricle, which collects the sound waves through the external auditory canal. It goes to the tympanic membrane, to the middle ear. The middle ear is going to vibrate the malleus first, then the incus, then the stapes. The stapes foot plate is oval, so it is going to push into the oval window, which is going to vibrate the endolymph, which is the fluid in the scaly vestibule, which is going to vibrate the perilymph of the scaly vestibule. So the vibrations then vibrate the vestibular membrane to vibrate the endolymph of the cochlear duct, because this is the membranous labyrinth. The vibrations go through the basilar membrane, which bends the hair cells against the tectoral membrane, which stimulates cranial nerve 8, which is going to go to the thalamus and then to the temporal lobe. Now, as sound waves travel, sound waves can go all the way up to the apex. They'll go through the membranous labyrinth, and then they have to have a way to exit. So sound waves and auditory transduction stops here, but sound waves need a way to exit. So as we talk about sound waves travel through the basilar membrane. They are going to go to the inferior chamber, which is going to lead out. So we would say this is scaly tympany. So the perilymph of the scaly tympany is going to vibrate until it gets all the way back to the round window. And the round window is how sound exits. So oval window is where it enters. It goes to the perilymph of the scaly vestibule, to the endolymph of the cochlear duct, which is the membranous labyrinth. It goes through the basilar membrane, which vibrates the hair cells. And then it needs a way to exit. So right here, we have the round window. Now the job of the round window is to allow for the sound waves to exit. So sound waves are going to vibrate the perilymph, and then they're going to exit through the round window. That is auditory transduction. So there's a really good video to basically show you this that's on YouTube that I posted that says great video to go through everything. Literally, when we talk about the cochlea, it contains the organ of hearing, which is called the organ of Corti, the organ of hearing. And the way that you activate the organ of Corti is you bend the hair cells against the tectoral membrane, which shoots electrical impulses to cranial nerve 8, which it's going to be to the cochlear branch of cranial nerve 8, which stimulates sound. Now, the sound waves do have to exit, so they're going to go through to the inferior chamber and out the round window. So that's literally the cochlea and auditory transduction. But when you say auditory transduction, you have to start with sound waves in the outer ear all the way to sound waves to the inner ear, to the cochlea, and how sounds or auditory transduction occurs. So when we look at the inner ear anatomy, first of all, you have to understand that there's always going to be 3 chambers. The Osseous Labyrinth is always going to be called scaly vestibule and scaly tympany. The Membranous Labyrinth is going to have different names. So the cochlea is used for hearing. The Membranous Labyrinth is called the Cochlear Duct, but the organ of hearing is the organ of Corti. Now, the next thing that I talked about as far as vibrations go, the scala vestibule has perilymph, which is going to vibrate. Then it goes through the vestibular membrane, then it vibrates the endolymph of the membranous labyrinth with different names to the scaly tympany and then out. So this is a good way to remember the direction. So the cochlea is for hearing. The name of the membranous labyrinth is Cochlear Duct. This is called the organ of Corti. So now I want to go over the vestibule. Okay, your vestibule is the center portion. Now, when we talk about the vestibule, it's going to have two membranous labyrinths that are swollen areas, and the job of the membranous labyrinth of the vestibule is literally for communication between the two. So, when we look at the vestibule has a membranous labyrinth called the saccule. So this is the saccule. The saccule is smaller, and it's the membranous labyrinth that helps us communicate with the cochlea. And then the larger one that's next to the semicircular canals is going to be called the utricle. So this is the name of the membranous labyrinth. There's two of them in the vestibule, the utricle and the saccule. So we're in the vestibule. There's a large one that's near the semicircular canals called the utricle that helps us communicate with the semicircular canals. And the saccule helps us communicate with the cochlea. So literally, you're still going to have three chambers. So you have a superior chamber and you're going to have an inferior chamber. So we have two membranes that make the membranous labyrinth. So the vestibular membrane is on top, the basilar membrane is on bottom. That doesn't change. But we have two membranous labyrinths. So we have the large utricle, which is going to communicate with the semicircular canals. And then we have the saccule, which is small. And the saccule is going to communicate with the cochlea. Y'all can take notes on those. I'm not going to have room to draw it out. This is the membranous labyrinth. Now, everything is literally the same. The scala vestibuli is on top. The scala tympani is on bottom. This is going to be known as the vestibular membrane. And the bottom is going to be known on the basilar membrane. Now, basilar membrane is always going to be where it's at. Vestibular membrane is always on the top. That's what makes up the top and the bottom, and it divides it into three cavities. You can literally still label everything the same. Scala vestibuli, scala tympani. Now, your vestibule is going to contain organs involved with static equilibrium. When you say static equilibrium, this is when you're sitting still only. So basically, it's going to help you sense head position. When the head is tilted. Now that can be like flexion. It can also be extension. It can also be lateral flexion. When the body is not moving. So literally when you're sitting down in a chair and your head is not moving. But I can take my head down and I know my head is going down. So when we talk about static equilibrium, we need to know that the organ involved with static equilibrium is known as the macula. So the macula's location is in the membranous labyrinth, just like the organ of Corti was in the cochlear duct. The cochlear duct is the name of the membranous labyrinth. So the utricle and the saccule is the Membranous Labyrinth that contains the macula. Now, the good thing about each one of these, the organ of Corti and the macula, is they all pretty much look the same. You just changed the name of the Membranous Labyrinth and the name of the organ. So I can literally draw it the same. It's on the basilar membrane, except for now, we're gonna call this the macula. Now, the hair cells involved, there's gonna be short hair cells and long hair cells. So still you're gonna have stereocilia. So your hair cells are gonna be floating around here in the endolymph. The hair cells are literally gonna be attached to cranial nerve 8. But we have short hair cells known as stereocilia, but you also have long hair cells. So I'm going to draw a long one and a short one. They're still hair cells. The long hair cells are called kinocilium. So you have hair cells with microvilli called stereocilia, which is going to be the short ones, and then the taller ones, which is called kinocilia. Now, the membrane that sat on the organ of Corti is called the tectoral membrane. But see, look, it has everything the same except for we're saying we have tall hairs and short hairs. The macula is going to help us with static equilibrium. So, the name of the membrane that sits on top of the macula is known as the Oto Lithic Membrane. Instead of the tectoral membrane, this is called the Oto Lithic Membrane. Now, Oto means ear, lithic means rock. So, on this material, this is a gelatinous otolithic membrane. On top of it, it's got calcium carbonate crystals or ear rocks that sit on top of it. So you have ear rocks. You literally have rocks that sit on top of this gelatinous material. Otoliths are literally ear rocks made out of calcium carbonate. Oto means ear, lith means rock, and these are your ear rocks known as otoliths. Now, they are made out of calcium carbonate. Now, this is very different because it is going to sense when your head is tilted. So, the vestibule contains the organ known as the macula, which is going to sense your head position when the head is tilted. Now, I have to draw this in order for you to see it. So, we're going to talk about bending your head forward. So, here's a little person right here. It's going to be a horrible picture. Okay, your head is going to bend forward. Here's your nose and here's your chin. Here's an eye. Here's the mouth. When you bend your head forward, what happens is this gelatinous otolithic membrane, so it's gelatinous, so it's jelly-like, that's what gelatinous means. This otolithic membrane is going to basically bend forward. So when you take your head forward, your macula, here's your macula, is going to stay where it's at. And your hair cells of the macula, which basically stimulate the vestibular branch of cranial nerve 8, is going to bend your hair cells. So what happens is when you bend forward, is this gelatinous material is going to lean forward and all the rocks are going to be pulled down by gravity. So your hair cells, which are going to be stereocilia, the short ones, and the long ones called kinocilium, are going to go with the gelatinous material. They're going to bend in the direction of wherever you move because the ear rocks are heavy and this jelly is gonna sag. And all the ear rocks are gonna help pull the head forward by gravity. So your ear rocks sitting on top literally are gonna make the gelatinous material sag because all your ear rocks are gonna basically move to the front. So that's how you basically activate, sorry about that, the macula to activate cranial nerve 8. So when the hair cells bend, the only way that the hair cells can bend is the Oto Lithic Membrane sags. And pulls the hair cells. This is going to stimulate the vestibular branch of cranial nerve 8. And you say, oh, I'm bending my head forward. So literally, as you bend forward, the Oto Lithic Membrane also bends forward, and that's how you know your head is bent forward. The same thing happens when you take your head back, or if you take it to the side, or if you take it to the other side. Your otolithic membrane sags, and the otoliths, or the ear rocks, just pull on that side, and then you know that you're going to the right, or you know that you're going to the left. But this is only when the body is not moving. So your vestibule, the name of the Membranous Labyrinth is called utricle. This is the larger one that communicates with the semicircular canals, and the saccule, which is going to communicate with the cochlear duct. But inside of your utricle and your saccule, you have the macula, which has hair cells that are going to bend, which are going to stimulate cranial nerve 8. So static equilibrium is going to happen in the vestibule because of the macula, which is located in the utricle and the saccule. So every time that we talk about each of the areas, you literally have to remember that the membranous labyrinth has different names, and it's going to have different organs that do different things. Now, they always are going to look the same. The organ of Corti has hair cells called stereocilia, and the tectoral membrane is what the hair cells bend against. In the utricle and the saccule, you have the macula, but this is going to have long and short hair cells that bend against a different membrane called the Oto Lithic Membrane, which move with gravity, and that's how we stimulate the vestibular branch of cranial nerve 8. So now we're on to the last one, which is in the semicircular canals. It's going to always be something like this. I just draw it like this because we focus on this inner section. All right, so we are on to the semicircular canals. Your semicircular canals are going to have organs involved with dynamic equilibrium. So one of the things that you need to understand is the difference between dynamic equilibrium and static equilibrium. Dynamic equilibrium is going to detect head movements with rotation or angular movements. Now, when we talk about rotation movements is going to be like when you're spinning a chair. That's dynamic equilibrium. So you're going to have balance when you're spinning in a chair. Angular movements is when you're sitting in a car you can feel that you're moving forward. So on a roller coaster, when you're moving forward, that's an angular movement. So angular movements are linear movements. So rotation, spinning, and angular is linear. So when you're moving in a roller coaster going forward, that's an angular movement. If you're spinning around in, I don't know what you could be spinning around in, like a teacup at Disney World or whatever, that's a rotation movement. That is dynamic equilibrium. So we always start with our 3 layers. So you have your scaly vestibule, you're also going to have your scaly tympani. You're still going to have your membranous labyrinth. So each one is going to have a different membranous labyrinth, but everything else is pretty much the same. So you can always say that the superior chamber is scaly vestibule, which is part of the Osseous Labyrinth. The bottom one is the scaly tympani, Osseous Labyrinth, which has perilymph. However, your membranous labyrinth is different in each one. So when we talk about the semicircular canals, there's 3 semicircular canals. So the membranous labyrinth and the semicircular canals, right here, and right here, there's 3 semicircular canals. These are known as the ampulla. So there's an ampulla here, here, and then a third one right here. So probably not the best color, but the membranous labyrinth in the semicircular canals is called the ampulla. Now, it contains the organs that are involved with dynamic equilibrium, which is going to be called the crista ampullaris. We can literally go through each layer and ask about the organ of Corti is for hearing and how it bends hairs against the tectoral membrane. Then we move to the macula, which is in the utricle and the saccule, which is responsible for static equilibrium, which is going to have hair cells. The Oto Lithic Membrane versus the tectoral membrane, but they look the same. Macula with hair cells, organ of Corti with hair cells, tectoral membrane, Oto Lithic Membrane. So this one, the ampulla, has the crista ampullaris, which is also going to look very similar to the macula and the organ of Corti. I draw it just a little baby different. All right, so the crista ampullaris. Also has hair cells that are connected to cranial nerve 8. So we have hair cells attached to the crista ampullaris. You have short ones called stereocilia, microvilli, and then you have kinocilium, which are going to be floating around here in the endolymph of the ampulla, stereocilia and kinocilia. Short ones and large ones. Now, your hair cells are going to be connected to cranial nerve 8. So, if the hair cells bend, then they're going to stimulate the vestibular branch of cranial nerve 8 because the vestibular branch is associated with equilibrium. Now, the membrane in the vestibule is called the Oto Lithic Membrane, and the membrane in the organ of Corti, or involved with the organ of Corti, is called the tectoral membrane. This one kind of looks like an upside down cup, like a wine cup or something like that. It's actually a little more pointed than this, but literally just think of it as an upside down wine glass. Now, we call this the cupula. So, the crista ampullaris is going to be responsible for dynamic equilibrium when the hair cells bend. Now, the cupula is the membranes that are going to move the hair cells, which is going to stimulate the vestibular branch of cranial nerve 8. So, let's talk about the cupula. When you're stationary, that means when you're sitting still, the cupula, I did not spell that right. The cupula is upright. It's literally just sitting there. Now, the only time that you're going to stimulate the crista ampularis is with rotation, which is spinning, or angular movements, which is linear. So literally moving in a go-kart going forward or moving in a roller coaster. Now, the crista ampularis is found in the membranous labyrinth. The membranous labyrinth is going to be called the ampulla. That is the name of the membranous labyrinth of the semicircular canals. Now, when you spin or you have rapid head movement, this is different than the Oto Lithic Membrane. The cupula actually goes in the opposite direction. So if you are spinning to the right, it's going to go to the left. So I always think of it like the cupula is like a gelatinous jelly that if you were carrying a plate with jelly and you spin to the right, it's such a jelly that it's going to go in the opposite direction. So the cupula is actually going to go in the opposite direction of whichever way you're spinning. So if you're spinning to the right, the cupula is going to move to the left and bend the hair cells. So when you have rapid head movement, which is spinning or linear, the cupula is gelatinous. The cupula bends in the opposite direction which bends the hair cells, and the hair cells are going to be called stereocilia and kinocilium. The kinocilium are the long ones. That literally is going to stimulate the vestibular branch of cranial nerve 8, and then you have dynamic equilibrium. So the key for the inner ear is understanding the membranous labyrinth all have different names in each of the areas, and they each have a different organ involved with different functions. So your crista ampularis is involved with dynamic equilibrium, but the way that it works is the hair cells bend as the cupula goes in the opposite direction. So if you're spinning to the right, the cupula goes to the left, and then that's how you know that you're spinning. If we went back, we always start with the inner ear. We're going to go through this one time. So the first thing that we went over is the inner ear. There's three things: the vestibule, the cochlea, and the semicircular canals. Now there's three chambers. We do call this the labyrinth or the maze. This is a maze. The Osseous Labyrinth on the top is always called the scala vestibuli. The vestibular membrane, the basilar membrane are the membranes that make the membranous labyrinth, and the bottom one is called scalae tympani. Your organs involved with dynamic equilibrium, static equilibrium with the vestibule, hearing with the cochlea, and dynamic equilibrium are always going to be in the membranous labyrinth, and they have different names. The cochlear duct is in the cochlea, the utricle and saccule is in the vestibule, and the ampullas are in the semicircular canals. They're always going to have a different name. The organs are going to have different names. So the first thing we did was the fluids, so that we could understand the cochlea and auditory transduction, which involves the oval window and the round window. The organ of Corti is the organ of hearing, and the way you stimulate the cranial nerve 8 is you have to bend the hair cells as the sound waves travel through the basilar membrane and bend against the tectoral membrane. In the vestibule, the way that you sense static equilibrium when your head is tilted, the Oto Lithic Membrane is gel and it sags and it bends the hair cells in the direction you're bending your head. Oto Lithic is rocks. The macula is how you do static equilibrium. The membranous labyrinth is utricle and saccule. The semicircular canals. The ampulla is the membranous labyrinth. The dynamic equilibrium is detected by the crista ampullaris, which has hair cells that bend, as the cupula, or the gelatinous material, goes in the opposite direction. So that is basically the inner ear. To me, this is a little bit easier than just reading through the PowerPoints to understand that they all look similar, but they're named different things.